Good day, class, and welcome to your next lecture from Mr. Moriarty and, of course, Spider-Man and Batman back there helping you out. And today is going to be all about the double helix and the dogma of biology. DNA, it is the be-all of life, and so it's probably the most important thing I think you can understand if you want to understand how life works and however these wonderful chemicals came together to form a way to transfer genetic information from one organism to the next. How they came to be in the first place is amazing, but eventually I'll get you into my theory of conscious evolution and how this molecule has arranged itself in a various way to create human beings who can consciously understand this molecule and have actually been able to start manipulating this molecule to create life and understand life and maybe even evolve life. So has that molecule come to the point where this chemical has evolved the ability to consciously change itself rather than just through random mutations of it? Really just mind-blowing thoughts there that I hope we can have time to discuss in the future. But before we can get to that really understanding, we got to start to understand this beautiful double helix that up until about the 1950s, this model was thought to be upside down, and nobody could really figure it out until Watson Crick stole some information from a lovely lady scientist named Rosalind Franklin, and they discovered the shape of DNA, but they were not the ones who discovered DNA or what it's made out of. They just discovered the shape of it. But by understanding the shape, we began to understand the code much better, which means we can now genetically engineer things. We can clone things, all kinds of wonderful stuff, thanks to the radiographs by Rosalind Franklin. So just a shout out to pretty awesome lady. We'll learn about another awesome lady in science later this week. So here we go into DNA. I'll get rid of this little window, kind of hide it away, and we will start with what we need to know about DNA. Today should be some review, but we might go a little deeper into the rest of it. I really like the background I have here showing the double helix, and then you can see the nitrogenous bases right here, which we'll get into shortly. To start, this is what we call the dogma of biology. If there's anything you get out of this year, it's to understand that DNA will write a code or transcribes a code into mRNA, and that mRNA is eventually translated into a sequence of amino acids that makes proteins. Proteins are really the be-all of the cell of any living organism. Proteins determine where the carbohydrates are used, how the carbohydrates are used, where the fats in fatty acids go to form phospholipid bilayers that make up the membranes of cells and membranes of organelles. It determines where things go in the cell, how things get put together. When you start looking into nervous cells, system cells, and the brain, it's these proteins and how they are arranged that even allow me to talk to you, to think about talking to you, to dream, to come up with ideas, to think logically. So proteins are the be-all of life, and to get to proteins, DNA tells us how to code those proteins and how that information can be stored and transferred from one generation to the next generation to the next generation. And so this is the dogma of biology. It is the code of life. So dogma, by the way, is a principle or set of principles laid down by an authority as an incontrovertible, I can't even say that word, true, but it is just the way it is. A lot of times dogma is used in religion because it is the way the origin is, and people said it. In this case, it's not a bunch of priests or holy men doing it. This is how life is. It is the science of life. And so really what this is the recipe instructions for all life is found in DNA, which tells the cell which proteins to build to make life happen. So protein synthesis is that dogma of biology. Where is my coding? I'm going to take a quick break. I need a drink of water, and I can't find my pause button on here, so we'll just come back. Maybe I'll edit that out. Oh, there's my pause button. Water, so important to life. Okay, let's continue on with the DNA, it makes the protein. Here is just a diagram of the awesome sugar that is found in the base of DNA. This is called a deoxyribose sugar. And you find these little sugars right here. And this is a kind of chemical diagram of how DNA is formed. Things we'll start learning is DNA runs in a 5' to 3' direction. 5' means this carbon is in the 5th place. 3 is the carbon here. So if you count the carbons on here, you have 1, 2, 3, 4, 5 carbons. And so molecules are oftentimes named by where the carbon is. So if you think back to triose phosphate, that's because in that carbon molecule, there would be 1, 2, 3. It would be on the 3rd carbon is where you'd find the phosphate. Don't need you to know that as much right now, but it will come back into play, so I want to talk about it. So this is the structure of DNA. We're going to go into it deeper, and it should be some review. You have this phosphate backbone that kind of forms the ladder of DNA. You have the sugar. All DNA and RNA have a sugar. They're just slightly different. And then we have these pink things are called the nitrogenous bases. And there are 4 different nitrogenous bases that you need to know. If you go further into biology, you actually find there's about 6 or 7 of these. And you see those more in cancer cells and stuff. But we're not going to worry about them. We're going to keep it simple with AG, CT, and U and RNA. Okay, so nucleic acids is the class of biomolecules that make up DNA and RNA. So our class of biomolecules, we have 4 of them. Carbohydrates, remember, are made of carbon, hydrogen, and oxygen. Fats, lipids, are made of carbon, hydrogen, and oxygen, although phospholipids have a phosphate in there as well. Proteins, they are what make us what we are. Proteins are made of chons, carbon, hydrogen, oxygen, nitrogen, and sulfur. And then we have nucleic acids as the last one. And I waited to talk about those until DNA because, well, it's the DNA. But they are made of carbon, hydrogen, oxygen, nitrogen, and phosphorus. Phosphorus because of that phosphate background. DNA is deoxyribonucleic acid. So it's a double-stranded nucleic acid made up of a sugar, phosphate, and a base. We just kind of looked at that. Deoxyribose is the sugar in DNA. I have a picture of that right here. Unlike most sugars we've talked about, it's actually a 5-carbon sugar. Makes kind of a pentagon shape. Phosphate is the backbone. So looking at this again, a slightly different drawing, we have our phosphate attached to a sugar, and these link the backbone of the DNA. So it's kind of like the ladder rung of a DNA. And finally, we have nitrogenous bases. There are 4 different nitrogenous bases, and these are the codes for DNA. So I think you remember back to freshman year or before, and we talk about DNA codes. You have the C, the T, the A, and the G, cytosine, thymine, adenine, and guanine. And then, of course, uracil is going to be in RNA. We'll talk about that later. Here's another look at it. So we have our phosphate backbone attached to our sugar, and then that would attach to one of these bases. And the sequence of those bases codes for proteins. So adenine, cytosine, guanine. These are the ones I want you to know. We can shorthand them most of the time to ACG. And I should have thymine on there. I really screwed that up. I'm going to try to fix that quick. Whew. Don't know how I did that. And that easy, fixed. Okay. Adenine binds with thymine. This should be review for you. A to T. It's actually bound by little hydrogen bonding, similar to how water bonds to itself. This is a very weak bond. And then cytosine binds to guanine. In this case, you have 3 hydrogen bonds that bond these 2 together. And as I said before, these hydrogen bonds are kind of weak. Okay. A nucleotide is a big vocab word I want you to know. That stands for a single unit of DNA or RNA. When free in a cell, that has actually 3 phosphates attached to it, like ATP. So we've talked about a nucleotide before. ATP is a nucleotide, adenosine triphosphate. And then here are the nucleotides for DNA, showing you when they're free in a cell, not attached to DNA. Instead of having just one phosphate, they actually have 3 phosphate groups. So these things are floating around in your cell when we're making DNA or copying DNA. They're there for all kinds of things. They're also floating around in the cell in the form of ribosomal nucleotides for making mRNA. And I just want you to look at these 3 phosphates and think to yourself, what do you think that phosphates are used for and why these would have those phosphates on them? And if we think about ATP, adenine triphosphate, wow, really it is the same thing that goes in DNA. It's because we can harvest energy for this. So when we're building DNA or building RNA, we need energy, and these come prepackaged with their own energy. So they got those phosphates groups on them, and we just chop these 2 phosphates off, and that energy is used to actually put DNA together and form strands of DNA. So it's kind of interesting, something you probably haven't thought about before, but wanted to bring up to you. So nucleotide, I'm going to use that word a lot. Nucleotide is a single base of DNA. So it does compose of a phosphate, it composes of the sugar, and then whatever base it is, but a nucleotide is considered like a single DNA, where DNA is the whole strand. Okay, so that is the basics of DNA. We also have molecules, nucleotides that are nucleic acids that are RNA. Big difference between the two is instead of a deoxyribonucleic acid, they have a ribonucleic acid. It's just got 1 less oxygen. Not really that much different. I don't care if you remember that much. But RNA is actually really almost cooler than DNA personally, because as we're going to find out, RNA does all kinds of cool things. But it's similar to DNA, has that ribose sugar instead of a deoxyribose sugar. And thymine is converted to uracil and RNA. So they're very similar chemicals. They have the same bonding. Hydrogen bonds can come off them, but uracil is a little shorter and just is used in RNA. Some of the reason uracil is in RNA is because thymine is not a very stable chemical and it changes very quickly. So once it leaves that double-stranded nature of DNA is almost always found in double strands, it can mutate pretty quick, and uracil is just a little bit better at not mutating. So that's probably why it evolved to be that way. There are going to be a bunch of types of RNA. There's some that I want you to know about. You were probably taught in freshman year or so that RNA is single-stranded. It is single-stranded. It can live single-stranded, but it can also fold into really cool shapes. And since it can fold into cool things, it can become a molecule that does a lot of different functions. RNAs can actually act like enzymes at times. There are enzymes working in your body that are actually made of RNA and not protein. Because of that, we think RNA might have been one of the more beginnings of life, like the start chemical evolution of life might have been little RNAs that formed in the primordial soup of life. And then DNA kind of came afterwards as a more stable molecule to store information. But RNA uses these same nucleotides, but they can actually do work. They can do a lot of things. So it's not just this simple single-stranded thing. It actually does a lot of cool stuff, and we'll talk about a lot of those. So the main types of RNA that I want you to know are messenger RNA. This should be review. We call this mRNA. We don't have to actually say messenger RNA anymore. We can just call it mRNA. It's going to carry the DNA code to the ribosomes to make protein. So here is one of the first major metabolic processes we're going to learn, and this is transcription in protein synthesis in the dogma of biology. And it is a messenger RNA is made. It's a complementary strand to the DNA, and this is going to carry the message to the ribosomes to make protein. So mRNA is messenger RNA. Talk about that one a lot. We also have something called rRNA, which is ribosomal RNA. So this is the RNA that makes up ribosomes that build protein. So if you remember back to freshman year or last year, ribosomes are the factories that make protein. So the mRNA comes into the ribosome. These next things we talk about, tRNA, are going to bring in amino acids that match the codes on the RNA, and we build this long strand of amino acids that eventually turns into a protein, which is the code for life. But what we rarely tell you about is these ribosomes are actually made out of RNA. Here's kind of just a drawing of how the RNA, you can see how it folds on itself all the way in here. These are the two subunits of RNA in a yeast. Pretty complicated, but this whole big structure is actually made out of nucleic acids and RNA. And due to those different shapes, actually can help push mRNA through and make proteins. So our RNA is ribosomal RNA. And then we also, the other one I need you to know is something called tRNA, which is a transfer RNA. And what it's going to is it's going to carry a specific amino acid based on the code down here. It'll have a specific amino acid it carries, and then it transfers those into the polypeptide chain of the amino acid, forming those strands of amino acid, and that is the final part of protein synthesis. As you can see, again, this is single-stranded RNA, but you can see how it can be double-stranded. It can form these looping patterns. Because of this, it can actually, in some RNAs, do like enzymatic properties. So RNA probably more complicated than we just always think of a single-stranded RNA. It's much cooler than that because it can fold and bond to itself. And notice C still binds to G, G still binds to C, so it's kind of just the sequence of RNA also depends on how it's going to make these loops and codes. Another type of RNA you don't have to know, not at least yet, but I will talk about it in the future. It's really cool, are these things called microRNAs and RNAis, which are interference RNAs. We actually had a student at Stanley Lake who did his doctorate on these and was these are like really kind of new. Within the last 10 years, we've learned a ton about these. We use them in medicine all the time. But what they actually do is that it's a way to control how genes are made. So when we get into protein synthesis and how genes code for proteins, we also have to think like we can't just make a ton of one protein all the time. We only need to make it when we need to make it, and we have to regulate how much we make. And these little microRNAs are really kind of cool. These are the microRNAs, single-stranded RNAs. And what happens is we make this mRNA that's going to go make a protein, but if I can make a complementary strand, so that means it has the opposite base pairs, it'll like stick to this RNA, and if it's stuck, it can't make a protein. So these things are really cool. There's a new drug out there, it's been out for a couple years now, that actually people can use to make sure you don't get HIV if you're positive for it. And the way this drug works, I can't think of the drug. At least the brand name of the drug. I don't know. But people who are high risk for HIV can now take, what they've done is the RNA that makes a certain protein, that if you have that protein, you can get HIV. If you don't have that protein, you can't get HIV. It actually gives you a small strand of complementary microRNA that binds to the mRNA and makes it so your body no longer makes that protein, and it makes it so you actually cannot catch HIV. So there's tons of medicine coming out with these things, all about regulating genes. So we will talk about that in the future. You don't have to know about it now, but just so you know that there's quite a few different types of RNA in our bodies, in the cells, that do amazing things as you go further into the abyss almost of how proteins are made and this dogma of biology that is just unbelievable. And we're going to work with some of that because we're going to genetically engineer some bacteria at some point, or some of you already have. So that is the basics of the fundamentals of what DNA is made out of. We'll review that a little bit as we get into other parts. But the other part I want to talk to you today about is the difference between eukaryotic and prokaryotic DNA. So little reminder, prokaryotes are single-celled organisms called bacteria or archaea and they are much simpler than eukaryotic cells, which are eukaryotic cells. They're the ones with all the cell organelles. They also have a nucleus and compact DNA. And this is a micrograph, so a photo of DNA inside a bacteria here, prokaryotic, and the difference of a eukaryotic DNA here. In both cases, the DNA is really wound up tight. But here you can see it's wound up even more tight, and it forms this stuff called chromatin, really condensely packed DNA. Here it's still super coiled and compact, but not quite the same. So in prokaryotic DNA, this is just a very simple representation. DNA can be found in two things. We have the genotype DNA, which is called a nucleotoid. It has no membrane around it or anything, so it's free-floating DNA, and it's tightly compacted. And so this is like the genotype of the bacteria. But they can also have these plasmid DNAs. We're going to work a lot with those when we start to, if you're genetically engineering things. And this is just secondary DNA that can actually be passed from one bacteria to another. And it can code for different genes as well. Antibiotic resistance comes into play here a lot. And it's a circular DNA, so the DNA forms a circle, but much, much simpler than prokaryotic DNA. When we talk about replication and making of proteins, we're going to focus more on the simple prokaryotics rather than eukaryotics, because eukaryotics are pretty complex for an intro to biology. But we do have to know the differences between prokaryotic DNA and eukaryotic DNA. And so today we're going to talk more about eukaryotic DNA. In eukaryotic organisms, DNA is housed in things called chromosomes. I think we've talked about chromosomes before. You probably have a general idea. We normally see them here. This is one chromosome. This is another chromosome, or chromatids. They're tightly packed together, DNA, and we call them like super packed. But if you were to unravel this, you get DNA. So this picture kind of shows it unraveling as we go back till you get down to this simple stranded DNA back here. And we're going to get more into that. So DNA packaging is how we, in eukaryotes, condense this down into a very tight space, and that helps protect the DNA as well, so it's kind of important. And so some vocab we need to know is chromatin is going to be densely packed DNA. So this purple stuff here, this is chromatin. It's very packed DNA, and it's not just DNA, it's actually packed with proteins too. So we'll get into those. Histones are those proteins. They're positive charged proteins. DNA is negative, and so they kind of are attracted to each other, and the DNA actually is going to wrap around these histones to help tighten the DNA and what we call like supercoil it and turn it into chromatin. We also have H1 proteins. These are going to help with that wrapping and supercoiling of chromosomes. And then we have something called a nucleosome, which is going to be a group of 8 histones packed together with DNA wrapped around them, and it just helps compact that DNA more and more. Here's a picture of that. So I have my histone, or these red proteins. The H1 proteins here kind of lock them in, and the DNA actually coils around these histones twice, and then you get another histone. And so when you get 8 of these all wrapped in really tight together, we call that a nucleosome. You can even go further, and then of those 8 packed nucleosomes, you get like 30 more, and we call it 30S. I'm not going to worry about that. I just want you to have an idea that the DNA wraps around these histones really tightly, really compactly. And so here's kind of showing single-stranded DNA. Now it's wrapping around the histones. Here's the nucleosomes. So that's 8 of them wrapped up. And then you get those combined together, and that's how you wrap into a chromosome and get it really, really tight. And why this is important is because when DNA is really wrapped around here tightly, it can't be turned into a protein. There's no way for things to get in there. It's hidden, it's compact. And if you think about a cell, and your cell, like picture a skin cell, it has all the DNA to do everything that your body needs to function as a human being. But a skin cell only has to do one job, and that's to make skin. So you don't want to use any of the genes that are tightly packed in here in a skin cell, because you might start doing something you don't want. And that is where cancers can come into play, is when certain genes get turned on that shouldn't be turned on in certain cells. So this is one way we can regulate what proteins are made by a cell and differentiate how cells work, is because this tightly packed DNA does not code for a protein. In some cases, if we want to make a protein, little proteins will come in and it'll open up an area of DNA so that we can make a protein and get that information from it. So I like to think of DNA as like a library with all the information needed to build the living organism or the living cell, but not every cell needs to access all parts of the library all the time. So when you need to check out a book, we can unwrap a histone and check out that book for what protein you need to make. So as we go further into this and we talk about gene expression, like when genes are made, turned off or turned on, a lot of that happens when we uncoil these DNA around histones so that we can access them. And it's a pretty complex process. Everything you do, whether it's taking a drink of coffee, if you ever do drugs or anything, you actually change the way your DNA expresses itself. Even breathing does, so kind of interesting. Here's just kind of a really cool, once again, because we have these awesome technology and these electron microscopes. Here's actually a picture of DNA being compacted. So here's it's totally compacted, but here you can actually see the little histones forming and wrapping around in DNA, and I just think that's kind of cool. Next thing I want to talk about are types of DNA sequences in eukaryotes. We will also do prokaryotes, but they're much, much simpler. So here is an example of my first one. We have parts of our DNA that code for proteins. This is the dogma of DNA. These are probably what we'll focus on the most, and they're the most important. So these are what we call genes. So you have your DNA, and then all of a sudden you have a gene. Every gene is going to have something called a promoter sequence. So these are going to be sequences of DNA that tell certain proteins in your cell to attach here and start the production of a protein or copying of mRNA. So then you have your coding sequence, and then you also have a part that tells it to stop copying the DNA. And then we also are going to have a bunch of junk DNA outside of the genes. Okay. In eukaryotic cells, we have exons and introns. So exons are what actually code for the protein. So in this RNA coding sequence, we're going to have introns and extrons. Introns and exons. Exons are what actually makes the genes. Introns are just kind of wasted space in there in eukaryotic cells. In prokaryotes, all of it is exons. Everything goes into making a protein within a gene. Not the same in eukaryotes, so much more complicated. So 2% of your DNA is exons, 24% is DNA, and that's just in protein coding genes. So other parts of your DNA, we're going to be doing a lab on this, is highly repetitive sequences. We really don't know what they do, but they make up about 45% of your DNA. And as we learn more about DNA, we're learning that these actually do have some importance to us, but most of our DNA is kind of junk DNA. Some of that's probably important because DNA has mutations, and so if mutations occur here, who cares? They don't really affect how the organism works, so we're lucky when we have DNA mutations there because they don't really harm the organism. If you have a DNA mutation in here, that is not good. In a gene, we can have lots of mistakes happen if mutation occurs here. But if it happens here, not a big deal. Some really cool things I won't test you on too much is something called transposons. So this is areas of junk DNA that can actually jump around and change from one chromosome to a different, or change places on a chromosome. We think evolutionary. This is where we get new genes to form. Hemoglobin, actually, if we look at the history of hemoglobin through different organisms as it changes, we see a lot of these transposons where they actually move around. Humans don't have that many transposon regions anymore, but kind of cool. Down here just kind of shows junk DNA, and it shows repetitive sequences. So it's like GATA, GATA, GATA. You get all these just same repeats over and over and over again. Some of it is structural DNA. This is the stuff that's going to help form around those histones and things like that. Not going to worry about it much. But what we're going to worry about a little bit are short tandem repeats. So this is a short tandem repeat. And we use these for DNA fingerprinting. So most of human DNA is identical. Between me and you, we're over 99.9999999% the same DNA. because we make basically the same proteins and everything else. But there are slight mutations that have changed. And as we started to understand the genetic code more, we were able to identify that these short tandem repeats, there's ones that are very similar in most humans, but some humans end up having an extra repeat. So in this case, we have human 1, human 2, and human 3. And human 1 and 2 have the same 8 repeats, but human 3 has 9 repeats. So we have found these small regions of non-coding DNA where we actually have a lot of variation in humans. So even though we're so much similar, these areas typically have a lot of variation. You don't find a lot of variation in the genes, because if there's variation in the genes, probably means you have some kind of issue like sickle cell anemia or albinoism or something like that that's pretty obvious to be different. And so this is where we find our genes, and this is where we do DNA fingerprinting. So we use about 13 of these short tandem repeats that we know about. We're able to cut these out of our DNA and then compare them from like me to you, and that is how we do DNA profiling. So each of these dark bands are different tandem repeats, and so humans show a lot of variation here, and using those 13 STRs, the chances of somebody having the same 13 short tandem repeats are about 1 in a million or 1 in a billion. So pretty rare that you'd find somebody with those same repeats, and that is how we use that for genetic profiling. We're going to do this with viruses, at least in my senior bio class. IB, you guys won't be. Just a wrap up of all this, just the types of eukaryotic genes. Here are our exons. That's what codes for protein. Only 1.5% of your whole genome is exons. Regulatory sequences, we'll talk about, those are kind of part of genes, and it helps say when we're going to turn on and turn off a gene. And then you got introns take up a lot of that space, which is kind of, I think, areas that life has made where, hey, if we get a mutation here, I don't really care because it doesn't do anything. We might find someday it does. And then just all this other stuff. So here's those repetitive transposons that we don't really know what they do. I think as we learn in time, they probably have more to do than we think. They probably help regulate our DNA more than we'd ever believe. Just kind of an interesting idea that really it's only 1.5% of that DNA that really codes for what humans do. We are still discovering all the genes that are in the human genome as well. That is all for today. I am going to have a little assignment for you where you're going to do some research on how we discovered that it was DNA that is the hereditable code for life and not proteins, because proteins are really the things that make life work. And so for a long time, we thought proteins were probably the code, but it turns out DNA was. And I'm going to have you guys do some research on Avery, Hershey, and Chase on how they were actually able to discover in the 1950s that DNA is the code for life. And have a wonderful day. Don't forget to do your lecture questions, and I look forward to see you guys in class.